The digestive system. So the digestive system consists of all structures that involve eating of food, digesting the food, absorbing the nutrients from the food, and finally removing the ones that were not absorbed. So, in a summary, it's a food processing system of our body. Put that in mind. Now, during the course of this, I want you to take pen, write down every part that was colored, because those are the most important parts. Okay. The digestive system is divided into two segments, which is the alimentary canal and the accessory digestive organs. Accessory, assist, assistant, assist. Do you understand? So it's not actually a major part of it's not actually a part of the digestive system, but it is assisting the digestive system in different formats, which we are going to see. For the alimentary canal, that's the digestive tract, we have the mouth, the pharynx, the esophagus, the stomach, the small intestine, and the large intestine. While the accessory organs, the teeth, your tongue, the gallbladder, the salivary glands, the liver, and also the pancreas. Specifically, the liver, they will ask you what is the function of the liver, or which of these accessory organs is responsible for the bile production. Please remember, bile production is liver, storage is gallbladder, production is liver, storage is gallbladder. Now you need to know why I shaded the accessory organs is because you need to know them. If you don't know the accessory organs, how will you know which one is not part of the accessory organs? Because they will ask you, one of the following is not part of the accessory organs. Do you understand? Good. Now, I noted down that the digestive system processes food, extracts nutrients, and eliminates residue. This is one of your questions. They will be like, dash, processes food, extracts nutrients, and eliminates residue. That's the digestive system. Mind you, I'm only, trying to help you prepare for your exams. This is just a rapid review, not substituting for your textbooks or substituting for your slides or everything. No. But this is just a rapid review. So I need you to be focused. I need you to take your pen, paper, keep it beside you, and write down things. Pause when necessary because I speak very fast. So try to pause when you can. Now, these are the accessory digestive organs, and these are the main GI tracts. All right. So this one, you need to look at this very well because they will give you this picture and they'll be like, they'll point somewhere here. They'll be like, is this part part of the accessory or the major digestive tract? So you have to know what part is this. So how food moves down. This is the stomach, then the small intestine. From there, it enters the large intestine, sigmoid colon, then the anus. All right. So you need to know this trajectory, and you need to know that this supports, this supports, this supports. This one supports, this one supports, the tongue, the teeth, everything supports. But this is the main places food moves. Food does not move into the liver. I don't know if you understand what I'm saying. So I need you to, be very focused. Now, this is also something that you need to know before we even start this discussion properly. Now, the food goes inside your mouth. That is ingestion. Do you understand? Now, in the mouth, the mechanical digestion and the chemical digestion occurs. In the stomach, the mechanical digestion and the chemical digestion occurs. In the intestine, there is absorption. You see? Absorption in the intestine. Why in the large intestine, the chemical digestion does not occur there? Food just moves out, compaction. You understand? So this is very essential for you to know. Now, one of the more important things you need to know is the function of the digestive system. The function of the digestive system. They include ingestion, secretion, mixture and proportion, digestion itself, absorption, compaction, and defecation. I pointed out digestion and absorption because they're very important. As I mentioned prior to this slide, I said that the digestion part involves the mechanical and the chemical breakdown. So they will ask you, the breakdown of food into nutrients includes dash and dash, mechanical and chemical. And they will ask you, what is it? Which function does it serve? Digestive function. They will tell you the entrance of nutrient molecules into the bloodstream or body's own tissue is called what? Absorption. Or they may tell you one of the following is the definitions of absorption. Then they will put this as one of the options. You need to understand especially these two, the digestion part and the absorption part. Also know the compaction. Compaction is when food in the sigmoid colon, in the colon is, the fluid is extracted from it. So it becomes stronger. That's why the more you don't go to toilet, the more constipation you may have. You understand? Your physics will become so strong. So defecation is removal of ingestible remains. Now, when you're talking about mechanical digestion, you have to include these three things in your head: CCG or GCC, any way you want to go, So this is cutting, this is grinding, which happens in the teeth, and churning, which happens in the stomach. Cutting and grinding happens in the mouth, churning happens in the stomach. Pay attention to this. Now, where does mechanical digestion start? In the mouth. Where does chemical digestion start? In the mouth. Does mechanical and chemical digestion happen in the stomach? Yes. Does it happen in the large intestine, chemical digestion? No. You need to take down pen and write this, pause it, and write down if necessary. Now, when you're talking about chemical digestion, you're talking about hydrolysis reactions. What are hydrolysis reactions? They are reactions that involve water, lysis, breakdown. So when you pour water into a certain polymer, for it to break down into smaller monomers, that is hydrolysis reaction. It is very important for you to know that chemical digestion starts in the mouth. I mentioned it again. I'll keep repeating this until it sticks in your head. Now, there are digestive enzymes like salivary gland, stomach, in the salivary gland, the stomach, the pancreas, and the small intestine. But what I need you to understand is that chemical digestion breaks down large polymers to smaller monomers, as I mentioned. Now, what are the polymers? We have polysaccharides, proteins, fats, and nucleic acids. Polysaccharides to monosaccharides, protein to amino acids, fat to monoglycerides and fatty acids, and nucleic acid into nucleotides. These are polymers. They are large. The body cannot take them in. That's why they are polymers. They are too large. So they have to be broken down. But what about the ones that we take in that are little, that the body does not need to break down? You need to know them. They include the vitamins, the amino acids, the minerals, the cholesterol, and water. Your question will come from this. They will be like, vitamins, they will be like, one of the following is ingested without breaking down. You understand? One of the following is ingested I'll take that again. So the way your question will come, they'll be like, one of the following does not need to be broken down, for it to be absorbed. Or one of the following is absorbed directly after ingestion. You need to look at vitamins, amino acid, minerals, cholesterol, and water. You understand? So, but note that all these ones are too big. They have to be broken down. But these ones are little. They don't need to be broken down. Now, moving to the GIT structure, there are 4 structures you need to put in your head: mucosa, submucosa, muscularis externa, and serosa or adventitia, or mesentery. Okay, now, mucosa is the innermost, serosa is the outermost. I need you to understand it that way. Then the submucosa, which is after the mucosa, contains something very important, which is a neural control. You understand? We call it the submucosa plexus. Another name for it is miasnas, submiasna. So submucosa plexus, another name for it is miasna, submiasna. Do you understand? Because this sub has an here, which continues this miasna. But for the muscularis layer, it has muscularis myenteric, myenteric. Do you understand? So the muscularis layer contains the myenteric plexus, which is also known as Auerbach's plexus. You have to put this in your head because they may ask you, the muscularis layer contains which plexus? You have to know. And in the option, they will put Auerbach's. They will not put myenteric. So you need to know that the muscularis layer has the Auerbach's plexus, also known as the myenteric plexus, while the submucosa has the submucosa plexus or the miasnas plexus. Then we have the serosa or adventitia or mesentery, which is the outermost part. Now, I need you to look at these because you need to know the sequence, how they follow. This is the mucosa, the innermost layer. The mucosa contains the epithelium, lamina propria. This is it. This. Then the muscularis mucosa. After that, you come to the submucosa and you have the submucosa gland, the blood vessel, and also the submucosa nerve plexus. That's miasnas plexus. You need to know this. Now, after that, you go to the muscularis externa. That is the muscle. The first, the innermost is the inner circular layer and the outer longitudinal layer. In the stomach, we also have the one that is diagonal. We will see it when we get to the stomach. But you have to know that for the submucosa, this is the plexus. submucosal plexus or Mesenteries plexus. But for the muscularis, this is plexus. That's Mesenteric plexus or Embark's plexus. You cannot fail to know this. All right? So we've discussed the mucosa. You understand? The mucosa is usually non-keratinized, stratified, squamous, epithelial, some parts, while the other parts are the columnar. We will still discuss that. Now, the submucosa consists of areolar connective tissue that binds the mucosa to the muscularis. But what I need you to remember for the submucosa is that it contains many blood and lymphatic vessels that receive blood, absorbed food molecules. Mind you, the submucosa contains many blood and lymphatic vessels, and this helps to receive absorbed food molecules. This is your exam question. This is very important. Another important exam question is that the function of muscularis externa. Put it in your head. Know the function. What does he do? Muscle. What does muscle do? Contract. Responsible for motility and propulsion of food and residue through the tract. Very, very, very important. In some parts, like the mouth, pharynx, and the superior middle parts of the esophagus, it is skeletal muscle. While in some parts, which is the rest of the GI, it is smooth muscle. You need to know this. Discuss about Mesenteric plexus. Another thing you need to know about is the sphincters. This muscularis externa also helps to serve as sphincter. What is sphincter? Sphincter are valves that help to prevent backflow of anything in the blood, in the GI, like in the heart. We have valves. We don't want blood to go back. When anything goes back in the body, it is a pathology many times. So when blood goes back, it becomes pathology. That's when you start thinking about heart failure. Do you understand? But in the digestive tract, it can lead to vomiting. So we don't want backflow. That's why we have sphincters. And those sphincters are by the muscularis externa. Bear that in mind. All right? Now, when you're talking about the serosa, another name for the serosa is the mesentery. Put it in your head. Do you understand? They will tell you that DASH is a serous membrane. But know that serosa is a serous membrane. But what you need to identify serosa with is the thin layer of areolar connective tissue and also the simple squamous epithelium. If they put these two, know that they're talking about the serosa. Just pick the serosa. Focus. They can tell you serosa, they can tell you mesentery, or they can tell you adventitia. You have to be cautious so that they don't confuse you with the question. But if this is what is going to come out, it is what is going to come out. Also know that the mesentery or the serosa or the adventitia helps to suspend the stomach and intestine from the abdominal wall. All right? So they hold everything into shape. There are some they pin to the abdominal wall. There are some that they are engulfing, which will lead us to yet another part, which is the retroperitoneal and the intraperitoneal. Retroperitoneal and intraperitoneal. Now, but before we get to that, I want you to look at these because this is the sequence from the innermost to the outermost. The epithelium, the lamina propria, the muscular mucosa, submucosa, muscularis externa, and the serosa. This is how they will tell you to arrange it in sequence. You have to note this. All right? Now, we are coming back to it. When you peritoneum, now the peritoneum is actually from the serosa. It's a double sheet, all right? Now, the peritoneum has the parietal peritoneum and the visceral peritoneum. The parietal is the one that is outermost, while the viscera is the one that's holding the organ itself. All right? So this is the visceral. You see the visceral, and this is the parietal peritoneum. This is peritoneal cavity. You don't really need to go too deep into it, but just know that it's divided into these two. We will discuss more about the peritoneum. Now, the peritoneum holds organs, correct? Now, there are some organs that is wrapping completely, and there are some that is just holding on one part. If it's wrapping an organ completely, it's called intraperitoneal. Intra. Do you understand? So those organs are intraperitoneal organs. What are they? Stomach, liver, parts of the small intestine, and large intestine. They will tell you one of the following is an intraperitoneal organ. Remember stomach and liver, very important. When they are telling you retroperitoneal is the ones that are held in the anterior part, pinning them to the posterior abdominal wall. All right? Examples of retroperitoneal is the duodenum, the pancreas. These retroperitoneal organs and some parts of the large intestine. Now, that same peritoneum gives birth to omentum. When it is connected to the stomach, it is omentum. It can be greater omentum or lesser omentum, depending on which curvature of the stomach it is arising from. If it's arising from the lesser curvature of the stomach to the liver, it is the lesser omentum. If it's falling up from the greater curvature of the stomach, it is the greater omentum. So if they tell you peritoneum that is attached to the stomach, know they are talking about omentum. But if it is not attached to the stomach, know that they are talking about mesentery. All right? Now, let's continue. Now, these mesenteries could be anywhere. Most times they are in the colon. If they are in the transverse colon, they are called transverse mesocolon. If they are in the sigmoid, they are called sigmoid mesocolon. If they are in the appendix, they are called mesoappendix. All right? So these are the things that will come out. That is why I shaded them. Another thing you need to know about the omentum is that the omentum is the part of the body's first defense against toxins and infections. Very important. You cannot forget this. All right? Now, let's see the diagrams of omentum. You see, this is lesser omentum. This is the stomach. This is the lesser curvature hanging from the stomach to the liver. Why this is the greater omentum? Extremely important. This is the parietal peritoneum. Know this and know peace. And most of these are the visceral peritoneum because they're holding the organs. Extremely important. I don't want to dive too deep into it because you are not doing masters in anatomy. You understand? So these also are areas that we consider, very important. That's why I put this picture. Now, another thing you need to know is that for digestive tract regulation, there are three. You can't forget. Three of them. One is the neural, the second one is the hormonal, third one is paracrine mechanism. So these three mechanisms are very, very important. They will give you an exam. Dash, dash, and dash are the mechanism that leads to motility and secretion of the digestive tract. It is neural, hormonal, and what? Paracrine. Now let's discuss these three of them. The first one, neural. We've discussed neural, funny enough. Neural is enteric nervous system. Enteric. It is independent. It works on its own. It is its own brain. Put this in your head. It's its own brain. You understand? So that's why we say independently of the central nervous system. So if they ask you, does the enteric nervous system work independently of the yes. How many years did I mention? 4 yeses. So as we proceed, also know that the enteric nervous system is now divided into two, which is some mucosa plexus I've been shouting about, and the mesenteric plexus. But one thing you need to know for me is know the function. First, you know the location. Anatomy is what? Location. Physiology is what? Function. For know the location, know the function. Know the anatomy, know the physiology. For the submucosa plexus, we have, they are found in the submucosa. Another name for it is meisner, correct? Now, what do? Secretion, mucosa, mucus, secretion. But for the mesenteric plexus, what do? They are found in the muscularis externa. So what does muscles do? Contract. So they are used to control peristalsis and contraction. Now, they are usually parasympathetic ganglia. Remember what I told you about the parasympathetic ganglia? If you've not watched my urinary video, please go and watch it. Please leave a comment also on this video, like also, and also subscribe. Don't forget to do that. Okay. Now, going back to parasympathetic ganglia, which mesenteric plexus is a component of. I told you that parasympathetic ganglia helps to relax the body, calm, to rest. But when you want to rest, that is when you start feeling like pooing. When you want to rest, that's when you start feeling like urinating. Meaning that the parasympathetic nervous system helps to do what? Control or increase digestion and also urinary system. I don't know if it makes sense. So this is neural control. The second one is hormonal control. The major important hormonal controls are gastrin and secretin. Very important. Put them in your head. Gastrin and secretin. They are chemical messengers secreted into the blood, and they help to stimulate the digestive tract. While paracrine secretions, just around the cells where they are functioning, that's where they are produced. Example is somatostatin and histamine. These are just succinct examples, but put them in your head. If they tell you one of the following is a paracrine secretion, somatostatin, histamine, they are the most common. One of the following is the hormone secreted for the digestive system, gastrin, secretin. Very important. Now let's dive into the mouth. All right? Very important. Why? Because the mouth is the genesis of the entire thing. The mouth is where the digestion starts from. All right? Another name for the mouth is oral or buccal cavity. It includes the cheeks, the lips, the hard and the soft palate, and the tongue. These areas are very important. It's divided into the vestibule and the mouth proper. All right? Now, this is the diagram. I don't want to spend much time on this diagram, but recognize the palatine tonsils, the uvula, the soft palate and the hard palate. The soft palate and the hard palate. then the palatine raft. All right. We also need to recognize the papillae. Okay, let's see the papillae. Now, this is the papillae. You see, this is the papillae. This is the filiform, fungiform, valate, foliate papillae. Look at them here. I need you to know this. These are also very important components of the mouth. Now, there is something I need you to know, which is that the mouth is lined with stratified epithelium. It can be keratinized or non-keratinized. It can be what? Keratinized or non-keratinized. I will show you the parts that are keratinized and non-keratinized. But also remember that the lingual papillae bumps. If they ask you the following, which of the following is a bump found in the mouth? It is the papillae, lingual papillae, which is the fungiform, filiform, valate, and the rest. All right. Now, when you're talking about the keratin also helps, sometimes to harden some areas of the mouth. That's why we say keratinized. But the part that is not keratinized is the part that you should know, which is the soft palate. But the hard palate is keratinized. That's why it's hard. But the soft palate is not keratinized. The lips, not keratinized. Cheeks, not keratinized. Surface of the tongue and floor of the mouth, not keratinized. Very, very important. So the tongue is divided into three parts, the two anterior two third and the posterior one third, and also the roots. We will see it. Then it's divided into two surfaces, which is dorsal and the ventral. All right. Now, for that papillae, I want you to see the papillae proper. You see, this is the papillae. If you trace it down, this is where it is found. This is the filipapillae. This is the fungiform papillae. So filiform, fungiform, the foliate, and the valate. You don't really know, you need to know the locations of these papillae. You understand? If you are still in college. All right. Just know that the bumps in the mouth are called papillae. That's enough. All right. Now, for the tongue, the functions include speech, taste, moves food, deglutition. Another name for deglutition is swallowing. Remember, moving food is mastication. It also helps to test buds like bitter, sour, salty, and sweet. All right. Now, why you need to know the divisions of the tongue is because of the sensory innervation and the blood supply. So the sensory nerve supply includes for the anterior to third, which is anterior to third. It involves the lingual nerve and the caudate tympanic, which is cranial nerve 5 and cranial nerve 7. Very important. Then we have the posterior one third, which is the glossopharyngeal nerve. Then we have the base, which is the internal laryngeal nerve. Very important for you to know this. Very important for you to know this, especially this posterior that has only one, because they will tell you one of the following is the posterior supply. One of the following is the posterior nervous supply of the posterior part of the tongue. Remember, it's glossopharyngeal. All right? I should have shaded it. Now, for the blood supply, we have the lingual artery, tonsillar artery, and the ascending pharyngeal artery. The veins, we have the lingual veins. All right? You don't really need to, master this, but just know them for knowing sake, in case of if you mistakenly see them in the options. Now, the muscles of the tongue includes the intrinsic muscles and extrinsic muscles. These are the extrinsic muscles. Extrinsic and the intrinsic, you need to know them. Extrinsic muscles are the glossus, like lip gloss. Glossus. You understand? So if anytime you see glossus, know that it's possibly extrinsic. Palatoglossus, styloglossus, hyoglossus, and genioglossus. All right? But the intrinsic involves all these: the inferior, the transverse, the vertical, the superior, intrinsic muscles. All right? So these are the intrinsic muscles. These are the extrinsic muscles. Something else you need to know is the components of the salivary gland. Remember, there are salivary glands in the mouth. You have to know the types of salivary glands and the components, because that's one of your questions. Mucus, lysos, amylase, lipase, all these things are components of the saliva. They will tell you one of the following is not a component of the saliva, or one of the following is not found in the saliva, and they'll give you mucus, they'll give you lysosome, they'll give you amylase, and they'll give you something else. Choose that something else. All right? Now, I keep mentioning that carbohydrate digestion begins what? In the mouth. But protein digestion begins what? Where? In the stomach. Remember, anatomy, location. Carbohydrate starts in the mouth, but protein starts in the stomach. We will still see it. Now, let us talk about the muscles, the salivary glands. Know that there is intrinsic and extrinsic salivary glands. The intrinsic is all this: lingual glands, labial glands, palatal glands, buccal glands, and etc. All right? Now, the most important is the extrinsic. The parotid is submandibular, sublingual. Parotid here. All right? It even have pathology. And what's the pathology? Mumps. Usually leads to inflammation of that parotid gland. This place will be swollen, most times among children, and it leads to infertility. The pathophysiology, we can discuss it next time, but mumps leads to infertility. So if a patient comes to the hospital and you are, for infertility, one of the questions you have to ask the person, have this place been swollen before? Have you had mumps before? Very important. So I need you to know the parotid, extrinsic salivary gland, the submandibular, and the sublingual. The submandibular is found around the mandible, and the sublingual is below the tongue. No this, no peace. All right? This is the diagram. This is the parotid. You see? This is parotid. Then this is sublingual, and this is submandibular. All right? I don't want to waste much time here. Now, let's talk about the palate. The palate has the hard part and the soft part. Very, very important. The anterior 2/3 of the palate is the hard palate, while the posterior 1/3 is the soft palate. There's a reason why this is very important. I need you to see the diagram first. This is the hard palate, you see? And this is the soft palate. We have the nasal ones and the oral parts, the nasal and the oral. But just notice that for the oral part, this is the oral part, this is the soft palate, and this is the hard palate. Then for the nasal part, this is the hard palate, and this is the soft palate. Enters here. All right? Now, let's discuss about the palate because it will come out in your exam. Both palates covered by pseudostratified ciliated columnar epithelium on the nasal region. So remember I told you something about the pseudostratified ciliated columnar epithelium, usually anything that does with respiration. Respiration. In the lungs, you will see the same thing. All right? So the nose most times deals with respiration. It has to, filter things that's going through it. That's why it has goblet cells. That's why it has glands that are mixed with both mucosa and serosa. All right? So know that for the both plates, that's the hard plate and the soft plate that are found on the nasal part is pseudostratified. But the ones that are found on the oral part is non-keratinized, stratified squamous epithelium. For the soft palate and for the hard palate is keratinized. Hard palate is keratinized for the oral part. Okay, let's look at the diagram and explain this thing proper. You see, this is the oral part. This is the nasal part. All right? Oral, nasal. We said that the oral side, the soft palate is non-keratinized. You see? So this is non-keratinized squamous. But this is keratinized squamous. That's why it's hard in the oral. But here, it is ciliated, pseudostrati here. Don't forget. I repeated it for you to be able to understand. So let's go to the teeth, all right? Which is one of the accessory organs as well. So we have permanent teeth and we have deciduous teeth. These deciduous teeth, I don't need you to start cramming all these things. I will tell you what to know. And these are the permanent teeth. What you need to know is the movement, how it moves in terms of arrangement. This is enamel, this is gingiva. Enamel always comes first. The gingiva follows. Enamel comes first. The gingiva follows. All right? Now, the dentine, the pulp, the root canal, and the periodontal ligaments, and also the dental alveolus. All these things make up the teeth. Now, it's divided into the crown, the neck, and the roots. All right? Now, they will ask you, the tooth has exposed crown. Yes. Constricted neck. Yes. One more roots. Yes. Does it make sense? Exposed crown. This is the crown. It's exposed. Yes. Constricted neck. This is the neck. It's constricted. Yes. Roots. Many roots. You see? Many roots. You have to put that in your mind. Another thing you need to know is the hard yellowish tissue that makes up most of the teeth is known as dentine. Dentine. Know that. They will ask you. The enamel covers crown and neck. Enamel covers crown and neck. If you look at the diagram, this is the enamel. Don't you see? It covers the crown and it covers the neck. Covers the crown, covers the neck. Very important. Know that the cement covers the roots. Why the root canal, is a space, leading to pulp cavity in the crown. These are very, very important. I specifically noted these places because these are the places that will come out in your exam. All right? Now, when you need to know the teeth numbers. For the milk teeth is 20. Milk teeth is 20. Permanent teeth is 32. Milk teeth is 20. Permanent teeth is 32. Milk teeth is usually found in babies or young children. But permanent teeth is from 6 to 25 years. You start having permanent teeth. At 25 years, I think it should be complete. All right? Very, very, very important. Now, let's not neglect the pharynx. Very important pharynx. All right? The pharynx has the stratified squamous epithelium, you understand? And also the ciliated pseudostratified columnar epithelium, goblet cells. Why? Because this part is close to the nose, but this part is close to the esophagus. Now, it's clear to you that anytime you think about the epithelium of something that is involving respiration, think about ciliated pseudostratified columnar. Think about it. But if it's for the mouth, think about squamous epithelium. It can be non-keratinized. Make sense? Now let's go to esophagus. Very long, 25 cm in length. All right? 25 cm. There are constrictions, a lot of constrictions in the esophagus, and it is found in the neck, thorax, and abdomen. Very, very important. Remember that food moves down through it as bolus. All right? Now look at the diagram of the esophagus proper. You see, it is just behind the trachea. We really need to know this location, and the pharynx enters into it. The reason is because why we need to know this is because we can be given NG tube sometimes. If you don't know it, you will never be able to give NG tube. Do you understand? So this is how it comes down and joins the stomach. And the area where it punctures to join the stomach, is known as the hiatus of the diaphragm. This is the diaphragm. You see the diaphragm as it's covering the entire thing. This area it punctures is known as what? The esophageal what? Hiatus. Very important for you to know. Now, when you are swallowing through the esophagus, what should you know? You should be able to know that about 22 muscles in the mouth, pharynx, and esophagus. Very important. So muscles in the mouth, pharynx, and esophagus involve in swallowing, not just the mouth, not just the pharynx, not just in esophagus. These three involves in swallowing. I know why I'm repeating myself. Now, know that the swallowing center is in the medulla oblongata. It helps to coordinate anything swallowing. So two things you need to know here is that muscles in the mouth, the pharynx, and the esophagus involve in deglutition, which is swallowing. Anyone you see in the exam, remember they are the same thing, deglutition and swallowing. Now, the swallowing center is in the medulla oblongata. Very important for you to know this. Know it and know peace. Now, let's discuss about the phases of swallowing. Think about it. The oral. You take food, you put it in your mouth, your tongue touches it. In short, mere looking at it, you're already salivating, correct? But when you put it in your mouth, it becomes the oral phase. All right? The oral phase. So food accumulates in the oropharynx, in front of the epiglottis. But the epiglottis have to cover, the trachea so that food does not enter into it. So the food now moves around and enters the laryngopharynx. This point is very important, but what I need you to know here is that the oral phase is under voluntary control because you are the one that voluntarily decides to eat. Think about it that way. I decided to eat. Oral, voluntary. Oral, voluntary. Oral, voluntary. Very important. When you are thinking about pharyngeal and you are thinking about gastric, those two phases are involuntary. They are autonomic. All right? And remember, breathing is suspended. They will ask you, is breathing suspended in pharyngeal phase? Yes. Is breathing suspended in pharyngeal phase? Yes. But I also need you to know that there are things that are also involved in pharyngeal phase, which you need to pause this video right now and look at all these things. Very important. But for me, I will take breathing is suspended and the pharyngeal phase is involuntary. Now, the esophageal phase involves what? Peristalsis. Peristalsis. Do you control peristalsis? No. It goes on its own. It goes on its own. Enteric, neuro. It goes on its own. Even if you turn somebody upside down, peristalsis will still go because it doesn't. Gravity does not have effect on it. So peristalsis ensures you can swallow regardless of your body position. Regardless of your body position. So you see some movies, they will turn the patient out, the individual upside down. And they'll give you food. You will still swallow it. They'll tell you, oh, I'm thirsty. Give me water. You give the person water. The person will still take it in. How? Peristalsis. So you have to understand this very, very well. Pause your slide and write down these things. Now let's go to the stomach. From the esophagus, we've jumped into the stomach. Beautiful stomach. Stomach is just a sac. It's just J-like shape. You understand? And we mentioned that the stomach is intraperitoneal. Don't forget that. Intraperitoneal. That's the question that they will ask you. Now, the stomach has different parts. If you look at this diagram very well, you see that this is the cardia, this is the notch, this is the fundus, this is the body, this is pyloric antrum, and this is pyloric canal. This is the pyloric junction, which is the junction between the stomach and the duodenum. All right? If you look at it very well. Now, I want you to appreciate the lesser curvature and the greater curvature. Why? Because in this lesser curvature, what happens? Peritoneum hangs from here to the liver. We discussed that before. What is it called? It is called the lesser omentum. I told you, once it's touching the stomach, it's an omentum. If it's dropping down from this curvature downwards, it's called the greater omentum. Know that, no peace. Now, functions of the stomach. Mechanically breaks up food, liquefies it, and begins chemical digestion of protein and fat. So chemical digestion of protein and fat starts where? In the stomach. Starts where? In the stomach. In the mouth? No, in the stomach. All right? So it will drop the food in form of semi-digested food particle, in form of chyme. So most digestive food occurs as chyme. They're not for them to pass into the intestine. All right? Now, one thing you need to know is that the stomach has different parts, which I've already mentioned. I need to look at it later at leisure. All right? Has curvature. I've shown you the greater and the smaller curvature, the lesser curvature. Two ends. We have the esophageal and the intestinal end, the cardiac notch, and the angular incisori. All right? These are other features that you need to know about the stomach. But one thing I need you to know is the interior part of the stomach. If you look at the interior part of the stomach, let me show you the diagram. Look at this interior part of the stomach. What do you see? What is this? Ruge. Very rough. Why? Because it needs to give a space, for food to be digested. Food needs to move slowly. It's in the stomach. And also for contraction, to press that food, to obliterate it. All right? So it's very, very important for you to know this. Now, we are still coming to this about the cells or the glands that are found in the stomach, what they produce. We're coming to this, all right? Now, but I need you to appreciate the anatomy of the stomach. Look at it very properly. You will see the anatomy of the stomach. If you look at it very well. Okay, I mentioned initially that the muscularis layer is longitudinal, circular, and what? Oblique. Remember, in other parts, it's just the longitudinal and the circular. But here is inner circular, outer longitudinal, and also the innermost. These are the things I want you to appreciate. This is the cardia. This is the fundus. All right? This is the sphincter, so that food from the duodenum does not move back to the stomach. This is a sphincter. There's also a sphincter here, which we don't want food in the stomach to move back to the esophagus and cause heartburn or vomiting. Very important. Now, let us look at the cells or the glands or the cells in the stomach. Function, physiology. What are some of the cells? One is the mucus. Two is the regenerative cells. Another one is parietal cells. Do you understand? Another one is chief cells, endocrine cells, and the gastric juice. We are going to discuss all this. First of all, starting mucus cells secretes what? Mucus. Just learn that and know that and know peace. Mucus secretes what? Mucus cells secretes what? Mucus. Where is this usually found? Around the cardia and the pyloric. Let me share it. Very important. Around the cardia and the pyloric glands. Cardiac and the pyloric regions. That is where you see the mucus secreting cells. Mucus secreting cells. When you talk about the regenerative stem cells, the name has given it out. What does it do? New cells, it produces new cells to replace cells that have died. Cells die constantly. So it will push out those cells, produce new cells. So that's the function of regenerative, which is another name for stem cells. So we've discussed mucus, we've discussed regenerative. Another one is parietal. Parietal, the senior man parietal. It's usually found mostly in the upper half of the gland. What does it do? HCl. What does it do? HCl. What does it do? HCl. What does this H do? HCl, can denature, vitamin B12. So because of that, parietal cells also produces intrinsic factor. This intrinsic factor will gum to the vitamin B12 to bypass the HCl and go to the small intestine, disintegrate, and is absorbed. All right? It also helps in the hormone called ghrelin. So parietal cells, think about HCl, think about intrinsic factor. Very, very, very important. Now we have the chief cells, or matter of them all. The chief cells, the most numerous, that's why they are the chief cells. But what do? They produce gastric lipase and pepsinogen. Gastric lipase and pepsinogen. But this gastric lipase and pepsinogen, what is activating them? The HCl. So you need to know that chief cells produces this, and this is not active, so it's activated by HCl. Does it make sense? Remember, we've discussed the mucus, secrete mucus, found here, regenerative, secrete new cells, parietal, HCl, the intrinsic factor, which helps vitamin B12 absorption. So this is chief cells, lipase, pepsinogen, lipase, pepsinogen. Now we have the endocrine cells. What do they secrete? They secrete hormones, paracrine messengers. Hormones and paracrine messengers. These hormones and paracrine messengers helps to regulate the endocrine cells. The chief cells is the most numerous cells, all right? That's why they are the chief. What do they secrete? Gastric lipase, pepsinogen. Pepsinogen is inactive. Remember I told you about zymogens. Pepsinogen is a zymogen. It is not active. It has to be activated by the HCl. All right? The HCl is very, very important. But in as much as it's activating the HCl, the bad thing about HCl is that it can destroy vitamin B12. That's why intrinsic factor is produced by the parietal cells to link up to marry vitamin B12. Hold it tight. Pass the stomach. Go to the intestine and divorce again. And the vitamin B12 is absorbed. So remember how the movement. Now the enteroendocrine cells, what are they? They secrete hormones and paracrine messengers, which helps to regulate histamine. Remember I told you paracrine, somatostatin, histamine. So these things they help to regulate digestion. They can ask you what are the components of the gastric juice. First of all, know that the gastric juice are produced a lot. 2 to 3 liters per day, a lot of them. You are pouring a lot of them. So they are mainly mixture of water, HCl, and pepsin. Water, HCl, and pepsin. A lot of them are produced. Water, HCl, and pepsin. They will tell you what are the components of the gastric juice. Please remember, water is there. Gastric, stomach, HCl. Okay. Pepsin, yes. Does it make sense? So I need you guys to remember this. Now, another thing you need to know is, what does hydrochloric acid do? As I mentioned initially, it helps to activate a lot of things like pepsin and lingual lipase. They will ask you, what activates pepsin and lingual lipase? Please, it is HCl. Another thing HCl do is to break up connective tissues, plant cell walls. Break up connective tissues, plant cell walls. Please, pause this video, write it down. Another thing they do is to convert ingested ferric ions to ferrous ions. And also they help to destroy ingested pathogens. These are the functions of HCl. You must know them. You must know them. You don't have a choice. Now, I told you about the genes. Gene. Anything gene is inactive. They are called zymogens. I need to shade this because this is also very important. They are called zymogens. Examples of zymogen is the pepsinogen, pepsinogen. This pepsinogen is broken down by the HCl to form what? Pepsin. So I need you to know what zymogens are. These are some examples of zymogen like trypsinogens, chemotrypsinogen, chemotrypsinogen. So trypsinogen is broken down to trypsin, activated, while chemotrypsinogen is activated to chemotrypsin. All right? We'll discuss this later. What you need to know is the innervation and circulation of the stomach. The stomach is innervated by parasympathetic fibers from the vagus and sympathetic fibers from the celiac. Parasympathetic from the vagus. PV. You see? Parasympathetic from the vagus. But sympathetic is celiac. Sin. Sympathetic celiac. So the blood supply is from celiac as well. So I need you to know this nervous supply and the blood supply. They are extremely very important. So when food is usually entered into the stomach, so I've described what sympathetic does and what parasympathetic does, all right? But another thing you need to know is the phases of food in the stomach. We have the cephalic phase, we have the gastric phase, and we have the intestinal phase. The cephalic phase is talking about phases: cephalic, gastric, and intestinal. Now, I'm telling you that the cephalic phase is you just hearing that, oh, there's a nice meal coming, or you see a meal, or you perceive it. Everything starts working in your body. The vagus nerve gets activated. Immediately, it starts stimulating HCl through the parietal cells and also, encouraging the gastrin secretion immediately. So that's the cephalic phase. I repeated it because that is how it will come out in your exam. Now, the second phase is the gastric phase. What is the gastric phase? The food has entered the stomach. So immediately the food enters the stomach, what happens? Remember I mentioned the endocrine cells in the pylorus and the duodenum of the stomach. What do? They secrete the hormone called gastrin. And this gastrin produces gastric juice. They secrete the hormone called gastrin. Gastrin produces gastric juice. And what does the gastric juice do? The gastric juice helps to, improve the digestion of food. You understand? It improves the digestion of food. Another thing is that the food that you eat will cause stretching. It will stretch the stomach. Immediately the stomach is stretched, the short reflex is mediated, and that short reflex is the myenteric plexus. Myenteric plexus, the same thing as the Albach's plexus. All right? And after that, a long reflex, which is the vagus nerve. The vagus nerve. And all these things help to increase the pH of the stomach. So when you eat food, it will cause stretching, and the pH of the stomach, is increased. Two things: stretching, increasing pH of the stomach. Put this in your head. Another thing, enteroendocrine cells secrete what? Gastrin. So which phase is gastrin secreted? Gastric phase. Now, let's talk about the regulation of the gastric function, right? So the gastric secretion is regulated by chemicals. What are those chemicals? Acetylcholine, histamine, and gastrin. Acetylcholine, histamine, and gastrin. Acetylcholine by the parasympathetic nerves, histamine by the enteroendocrine gland cells, gastrin by the enteroendocrine G cells. All right? So acetylcholine, histamine, gastrin. These three things, HAG, these three things regulate gastric function. So if you see it in your exam, please pick these three things. Very, very, very important. The intestinal phase, all right? So this intestinal phase is very, very important. You understand? Because a lot of things happens in the intestine. This is when food gets to the intestine. First of all, before we look at this slide, I want you to look at this slide very fast. The intestinal phase, we have the cephalic, we have the gastric, and we have the intestinal phase. Remember that. We've discussed the cephalic, we've discussed the gastric, and now we are in the intestinal phase. As a summary, the cephalic phase, I told you, it is when you are seeing, thinking, or testing. But when the gastric phase, the food is already in the stomach, stretching, leading to gastrin and histamine. All right? Now, when you are talking about the intestinal phase, food is now dropping into the intestine. You understand? What happens? CCK, secretin, produce. Also, GIP. These are the three things that we want to think about, and which is something you should also think about. Same thing with intestinal gastrin. The intestine has gastrin. All right? So bear this in mind as we look this slide. You understand? So that you'll be able to pick what we are discussing. So first, we mentioned that secretin and cholecystokinin produced by the endocrine cells in the intestinal mucosa. Correct? Now, some food has peptides and amino acids in them. So what happens? This activates the G cells of the duodenum. These G cells of duodenum produces more gastrin, which is exactly what we call intestinal gastrin. I need you to understand it properly. Now, it produces more gastrin. And this more gastrin, remember, stomach acids and fats, all right, trigger enterogastric reflex. What happens? The duodenum will now send signals to the stomach by way of enteric nervous system to inhibit anything that is happening in the stomach, all right? And also to inhibit the movement of the stomach into the intestine, because we don't want acid in the intestine. And also, we want to give time, for the duodenum to work on the time. So because of that, we have to stop the stomach from pouring things into the duodenum. This is what these inhibitory signals do. I need you to understand it. Also, even the duodenum also signals the medulla to inhibit vagal nuclei. This vagal nuclei will stimulate sympathetic neurons. All right? This sympathetic neuron does what? Inhibits signals to the stomach. So everything slows down. The secretion of gastric juice slows down. The gastric motility slows down. And these slowdowns will lead to activation of the pancreas and the gallbladder. Why? Because we need enzymes from the pancreas. We need bile from the gallbladder, correct? Because bile contains bile salts specifically, and bile salts helps in absorption. I don't know if it's clear. So another thing that happens is that the gastrin secretion in the stomach declines. Also, pyloric sphincter contracts to limit chyme entering into the duodenum. So all these things are to reduce chyme from entering the duodenum, so that the duodenum will be able to work on the limited chyme in it has. Now, if this thing does not work, then there's a problem. We'll discuss it later in terms of pathology. Now, remember one last thing that we mentioned here. You see it, if you look properly, you see the GIP. What does the GIP stand for? GIP, the glucose-dependent insulinotropic peptide, GIP. This is produced by the endocrine cells, all right? Now, what does this thing do? It helps to stimulate insulin secretion in preparation for processing nutrients about to be absorbed by the small intestine. So I need you to know these things. GIP, that's why I shaded them. That's why I shaded gastrin. That's why I shaded the secretin and the cholecystokinin, and also shaded the stomach acids fast that trigger enterogastric reflex and what duodenum do in response, which is inhibiting signals to the stomach and also in stimulating sympathetic neurons. If you know this, you are good to go. All right. Now, for the blood supply to the stomach, we have the left and right gastric arteries, right and left gastroepiploic arteries, short gastric arteries, posterior gastric artery. If you look at this, that everything from the aorta to the cilia, then celiac trunk, then the cilia came here, divides. This is left hepatic, and these are all the supplies. I need you to take time to look at this. I'm not going into it right now, all right? Because I'm just giving this thing surface level so that you focus on what is going to come out in your exams. Now, let us go to one of the major accessory organs. Remember, we discussed other accessory organs like the teeth, the mouth, the cheeks, the tongue. Now, one of the major accessory organs is the liver. The liver. The liver is the largest gland in the body, not the largest organ, mind you, the largest gland in the body. Has 4 lobes, the right, the left, caudate, and the quadrate lobes, all right? It also has fissures, all right? It has different surfaces. This we are going to look, structurally as we proceed. Now, before we go to those structural anatomy of the liver, let us talk about the histology of the liver. What do you know about the histology of the liver? What is going to come out in your exam? Please know the hepatocytes, know the bicanaliculi, know the hepatic sinusoids, know the Kupffer cells. If you know these 4 things, you are good to go. All right? What is hepatocytes? Hepatocytes is the functional unit of the liver. Just the way we have nephrons for the kidney, just the way we have cells for a human being. You understand? Hepatocytes is the functional unit of the liver. Pay attention to it. It's involved in virtually everything the liver does. Metabolic, secretory, endocrine function. You're talking about hepatocytes. Canaliculi is just a duct. You understand? It's a duct where hepatocytes, used to, push bile. So when bile is produced, they are pushed through the bicanaliculi. Bicanaliculi, duct, where the bile produced by the hepatocytes passes. You need to know this. The hepatic sinusoids is just talking about capillaries, so I'm not going too deep into it. But the Kupffer cell, another name for Kupffer cell is stellate reticuloendothelial cell. Stellate reticuloendothelial cell, very, very important. Why? Because it helps to destroy worn-out white and red blood cells. But not only these things, they will ask you in your exam, what helps to destroy bacteria? It is the stellate cells or the Kupffer cells. What helps to destroy foreign? It is the stellate or the Kupffer cell. This is something you cannot mistake. You have to know it. The blood supply of the liver, you understand? We have the portal vein and the hepatic artery. All right? This supplies and drains blood. You understand? Venous drainage, you have the portal vein also and the hepatic veins. I'm not going too deep into the vessels. These are the lobes of the liver, which is something you should know. Remember, this is the gallbladder. This is the parts of the gallbladder, which we'll discuss later. But this is the left lobe, this is the right lobe, all right? This is spaces in surfaces in the liver, all right? Now, this is the lobes proper. Left lobe, quadrate, and right lobe. Left lobe, quadrate, right lobe, all right? So that is that for that. Also know the things that are bordering the liver. Superiorly, we have the diaphragm covering above it, diaphragm. Anteriorly, in front of it, we have an abdominal wall. Inferiorly, we have the stomach and others. And posteriorly, we have the esophagus and others, especially inferior vena cava, the aorta. Even the gallbladder is posterior. The same thing with the vertebral column and the diaphragm. Some remnants of the diaphragm. Laterally, it's still bordered by the diaphragm and the lower ribs. Now, something I need you to also know is the portal triad. They will ask you in your exam. What is portal triad? This is the bile duct, portal artery, and portal vein. This is the portal triad. Triad. Portal. Do you understand? So this is portal triad, this is portal triad, this is portal triad. You see how the 3 of them together. The bile duct, the portal artery, and the portal vein. Now, when they are talking about the functions of the liver, which is another thing they will ask in your exam, remember it's for carbohydrate metabolism, Remember, it's for deamination of amino acids. Remember, it's for synthesis of plasma proteins, and blood clotting factors. All right. Other things are a bonus, like protein metabolism, transamination, desaturation of fat. These are the ones that they really ask a lot of questions on. Other things they ask questions on is like vitamin A synthesis, secretion and synthesis of bile by metabolizing cholesterol. This bile that was synthesized is stored in the gallbladder, Also, methanol, product, very, very important for the liver. Now, what you need to know is composition of the bile. What does bile contain? All these things: water, minerals, or mucus, bile pigments, bile salts, and cholesterol. What is most important to me here? Bile salts. What is most important to me here? Bile salts. Why? Because it helps in digestion. I mentioned initially when I was talking. Okay. The gallbladder, going to the gallbladder now. The gallbladder is just a pear-shaped sac, helps where the liver stores what? The liver stores bile. It's just a little below the liver, and it's a part of the biliary tract. It has the fundus, the body or the main tract, and the neck, which is continuous with the cystic duct. We will see the diagram as we proceed. This is the size. This is the parts that we've discussed right now. This is the blood supply. This is the nerve supply. This is the lymphatic drainage. Know that the lymphatic nerve supply has parasympathetic and sympathetic, as it is with everything in the nervous system. These are the blood supply: cystic artery, portal vein. Okay, we've discussed this to store bile. Gallbladder to store bile. Gallbladder to store bile. Because remember, food enters the GI, stimulates cholecystokinin, CCK. You understand? So CCK will contract and release by the sphincter of Oddi. It will contract, release the sphincter of Oddi. You understand? That sphincter of Oddi is where the bile enters into the intestine. We will look at it in the diagram. What you need to know about the pathologies of the gallbladder is that, number one, there is biliary calculi. Biliary calculi. That's the major one we call gallstone, because it's very hard. It's very, very hard. But remember, mind you, the difference it has with others is that it contains cholesterol, bicarbonate, calcium carbonate, and bilirubin. Extremely important. Extremely important. These three things is what is composed of. You understand? That makes up the gallstone or the biliary calculi. When you talk about cholelithiasis, you understand? It's majorly cholesterol. That's why it starts with chole. So anytime you hear it, excess cholesterol. So they give you question. DASH is one of the pathologies that is as a result of excess cholesterol. Please, cholelithiasis. But if they tell you, a hard mass in the gallbladder or the bile ducts, please choose gallstone. They ask you what are the components of gallstone. Tell them cholesterol, calcium carbonate, and bilirubin. Is it painful? Yes, because painful obstruction of ducts is very, very painful. You will feel it. Sometimes it leads to jaundice. Sometimes it leads to poor fat digestion and impaired absorption of fat-soluble vitamins. Why? Because if these stones block the gallbladder, bile will not be coming out now. And if bile is not coming out, what will digest fat? Nothing. Because bile salts help to digest fat. For you to take care of this situation, we need to do lithotripsy. What is lithotripsy? We pulverize the stones without surgery using ultrasonic vibration. Very important. Very important. Now, when you are talking about the bile ducts, the most important part that I really want to focus on is the cystic and the hepatic ducts. Why? Because these two things come together to form the common bile ducts. All right? So there are other things that you need to know about it, which I will show you in the diagram. If you look at it, well, this is cystic duct. This is that common hepatic duct that I mentioned. This common hepatic duct is coming from the liver. You see? The liver produces bile, pours it here, stores some of them here. Then others, here, this cystic duct and the common hepatic duct from the liver joins together to form common bile duct. Common bile duct. In the exam, look at the question very, very well, because they'll tell you cystic duct and common hepatic duct. Tell them common bile duct. Look at it here. Do you understand? Now, this is the intestine. How do they enter the intestine? This common bile duct will come down and we merge with the pancreatic ducts. They will merge with the pancreatic ducts and they will now enter this place. This is the duodenum. Now, the hole that they enter this place is known as hepatopancreatic ampulla. Hepatopancreatic ampulla. What is guiding this hepatopancreatic ampulla is the sphincter of Oddi. I need you to look at this. You have to understand this. Very, very, very important. So this is what biliary system is about. I don't want to stress on it. Now, we're entering another accessory organ. What is it? The pancreas. What is it? The pancreas. All right? So the pancreas, just know that it's an elongated accessory digestive gland that lies in the abdominal cavity. This is just talking about the location, which is the anatomy. Now let's talk about the function, which is the physiology. The pancreas helps to produce exocrine and endocrine gland. Endocrine, exocrine gland. The endocrine gland is the glucagon and the insulin. They will ask you, why didn't I share this? The glucagon and the insulin. Glucagon and insulin. These two hormones produce the pancreatic islets of Langerhans. They may not ask you this, but they will ask you, what are the endocrine secretions of the pancreas? This is glucagon and insulin. If they ask you, what are the exocrine secretion? Exocrine secretion, tell them is the pancreatic juice. Pancreatic juice, which is produced from the acini cells. All right? So pancreatic juice is the exocrine, while the glucagon and insulin is the endocrine. I am hitting on it. This is the diagram of the liver. This is the diagram of the gallbladder. And this is the diagram, of the pancreas. You see? You see the body, you see the neck, you see the main pancreatic duct. You understand? See how it comes, joins the bile duct here, the common bile duct. This is the common bile duct. And this is the cystic duct. This is the common hepatic duct. Joined together forms the common bile duct, which joins with the main pancreatic duct. You understand? And both of them enters here. You see? This is where they enter, the duodenum. Okay. Now, the function of the pancreas, especially the pancreatic juice, which that's the exocrine, is that it produces a lot of things. You understand? Like water, enzymes, zymogens, sodium bicarbonate, and other electrolytes. So without the pancreas pouring the sodium bicarbonate in the small intestine will continue to be acidic. All right? So this sodium bicarbonate buffers HCl. It buffers HCl. Very important. It buffers HCl that is arriving from the stomach, thereby making the duodenum not to be too acidic, instead to be basic. So that's how important the pancreas is. Also, remember it produces zymogens. We'll still discuss this later. All right? Now, the pancreas, as I said, it produces zymogens. What are those zymogens? Trypsinogen, chemotrypsinogen, chemotrypsinogen. So this trypsinogen is secreted in the intestinal lumen, yes. Remember what I told you about the genes, gene. The genes are inactive. So when this pancreas produces this inactive, you understand, it's now converted to trypsin by enteropeptidase. So enteropeptidase converts trypsinogen to trypsin. Trypsin will now start having autocatalytic reactions because it's an enzyme. All right? Autocatalytic action means that auto itself. So the trypsin will continue activating other trypsin. This time around, they are no longer waiting for the enteropeptidase. So immediately the pancreas produces trypsinogen, it will be converted to trypsin by enteropeptidase, and the trypsin will now start converting itself. More trypsin. The same thing happens with chemotrypsinogen. So chemotrypsinogen is also converted to trypsinogen by trypsin. All right? Trypsinogen by trypsin. I need you may not be able to understand this. So let's look at, okay, before we move out of this slide, know that there are other pancreatic enzymes like pancreatic amylase, pancreatic lipase, and this. All right? Ribonuclease and deoxyribonuclease, which digests RNA and DNA. While amylase digests starch, lipase digests fats. All right? So before we look at this, I want us to look at other pancreatic enzymes include the pancreatic amylase, which digests starch, the pancreatic lipase, which digests fats, and also ribonuclease and deoxyribonuclease, which digests RNA and DNA, respectively. But these are not extremely very important for your level. What is important for your level is for you to know how the secretions are being regulated. How the secretions are being regulated. There are three things that are responsible for pancreatic juice secretion. All right? That is acetylcholine, cholecystokinin, and secretin. Just know it that way. Acetylcholine, cholecystokinin, and secretin. All right? Now, when you are talking about cholecystokinin, that's CCK, we know where it's produced. Secretin is released from the duodenum in response to acidic chyme. You understand? What does it do? It stimulates ducts of both liver and pancreas to secrete more sodium bicarbonate. So cholecystokinin stimulates pancreatic acini. You must remember that. CCK stimulates pancreatic acini to secrete enzymes, while secretin does what? Stimulates both the liver and the pancreas to secrete more sodium bicarbonate. All right. Now, let's go to the small intestine proper. We are now back to one of the major digestive tract. That is the small intestine. We are done with most of the accessories. Now, when we are talking about the small intestine, it's the longest part of the digestive tract. The longest part. This is the small intestine. Very long. See it? Very, very long. 6 to 7 centimeters long. It consists of the duodenum, the jejunum, and the ileum. The duodenum, the jejunum, the ileum. We may not go too deep into studying the entire, area of the small intestine. So we are focusing on the things that we need. And one of those things that we need is to know the things that increases the surface of absorption in the small intestine. They will ask you, if you look, these are villi, you see? These microvilli. If you look at this place, what do you see? You see rugae, just like in the stomach. You understand? You usually call them Papillae secularis. All right. So this is rugae, this is villi, or even then, if you look within, you see microvilli. And the fourth thing is intestinal length. So because the intestine is very long, your digestion will happen in a lot of places. It gives you time before food moves through that 6 to 7 meters long. The food would have completely digested. Same thing with the circular foods. Same thing with the villi, same thing with microvilli and others. So these are the things that affects the digestion of food specifically. Now, another thing you also need to know in the microscopic anatomy of the small intestine, there are a lot of folds which we've discussed. But these folds, bear in mind that this fold causes chyme flow in a spiral path. Do you understand? Leading to more contact with the mucosa. Do you get? Now, this will promote more thorough mixing and nutrient absorption. This is what you should write down. So in the stomach, because of the length, in the intestine, the intestine, because of the length, because of microvilli, because of the villi, all right, and others, the chyme flows in a spiral path. Because of it flowing in a spiral path, it will now have a lot of contact with the mucosa. This is what they will tell you in it. This will help to promote more mixing, thorough mixing, nutrient absorption. You can't fail to understand this. All right? We are gradually coming to the end of this. So just be focused, continue writing, and make sure you pause to write down these things because these are your questions. Another thing you need to know is that the villi or the villus is covered with 2 types of epithelial cells. We have the absorptive cells and the goblet cells. The absorptive cells, the enterocytes, the goblet cells secrete mucus. All right? You need to know this. They will ask you, a villus, you understand, is covered by what? It's covered by what? Remember, it is epithelial cells. They may ask you, what are the types of epithelial cells by a villus? Tell them goblet cells and what? Absorptive cells, which is enterocytes. Also, they will tell you a lot of things. For example, that the lamina propria contains what? Arterioles, capillaries, venues, and lymphatic capillary. And all these things, we call them lacteal. 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 All right? So lacteal contains the arterioles, capillaries, and venues, and also the lymphatic capillary. All these things are called lacteal. If you see lacteal in your exam, you should know what they're talking about because that is your question. Another thing you need to know about the microscopic anatomy of the intestine is that the brush border enzymes contained in plasma membrane of the microvilli. We mentioned the villi, we mentioned the micro. So for the villi, absorptive and goblet cells, all right? For the folds, this is what the folds does. This is the villi. In the villi contains this lamina propria, which contains arterioles, capillaries, venues, and lymphatic capillaries. So I would just say that this lamina propria contains the lacteal. The lacteal is contained in the villi, all right? So this is for the villi. This is for the microvilli. It contains brush border enzymes. Make sense? So these brush border enzymes, is in the plasma membrane of that microvilli, not just everywhere in the microvilli. What do the brush border enzymes do? They carry out the final stages of enzymatic digestion. The final stages of enzymatic digestion happens by the brush border enzymes, which is contained in the plasma membrane, where microvilli. That's the point. For this digestion to occur, the chyme must definitely contact the digestion, the brush border enzymes. You understand? That's what we call contact digestion. Also, we have something called intestinal churning. You understand? This intestinal churning is just actions that ensures contact between the chyme and the mucosa. All right? They will also ask you, why does the intestine need to contract? 3 reasons: to neutralize acid. Another reason, to mix the chyme with all the juices, the bile and the pancreatic juice. Then the third one, for proper digestion. All these things lead to proper digestion. This is the idea of intestinal motility. This is the idea of contraction of the intestine. All right? So if they ask you, what is the major action of intestinal motility? Tell them that it is to churn chyme and bring it in contact with mucosa for contact digestion and nutrient absorption, and also to move residue toward where? The large intestine. To move residue towards the large intestine. This is what they will ask you. They will ask you, the churning of food and bringing in contact with the mucosa for contact digestion and nutrient absorption is known as what? Intestinal motility. Intestinal motility. What are the things intestinal? One of the following is not the things that A, to mix chyme with intestinal juice, bile, and pancreatic juice. B, to neutralize acid. C, to digest nutrients more effectively. D, they'll give you blah, 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 blah. The answer is that blah, 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 because they say one of the following is not. But if they say one of the following is, please look for any of these. All right? Now, part of the intestinal motility is something we call segmentation. What is segmentation? Things are happening in different places at their own time. Do you understand? So if this is the length of the intestine, here, around the four, there'll just be a ring-like formation. It's just segments on its own, and it's doing its own thing. The other part does its own thing. The other part does its own thing. At that moment, things are not moving. They are just stationary, doing their own thing. That is what we call segmentation. And this segmentation is carried out by enteric pacemaker cells in Muscularis externa. They will ask you, segmentation is caused by what? Please remember enteric pacemaker cells. Where are they found? Muscularis externa. Now, when food has finished digesting, what happens? Segmentation declines and peristalsis starts. So peristalsis is more like movements. Segmentation is stationary. You need to get this. Now, as I mentioned, peristalsis is movement, colon. That is your question. What is the function of peristalsis? What is the function of segmentation? All right? So peristalsis, migrating motor complex. What does it do? Successive overlapping of contraction. And it will take almost 2 hours for this thing to milk chyme towards the colon. So remember, peristalsis pushing chyme towards the colon. Pushing chyme towards the colon. Why segmentation is all about, bridging, stationary bridging, or constriction, stationary, ring-like. That is what segmentation is about. You need to know these 2 things. Very, very important. Another thing we need to know about the intestinal motility, which is peristalsis specifically, is that the peristaltic wave is triggered by motility. Know this and know peace. Just the way I told you that segmentation here, if you notice, segmentation here happens by the enteric pacemaker cells in the Muscularis externa. If you look down here, you will see that peristaltic wave is triggered by what? Motilin. They will ask you this or they will ask you that. All right? Motilin. And it takes almost 2 hours for all these things to happen. Now, function, physiology. Let us do a summary of this digestion before we go to the large intestine. Let us do a summary of it, like a recap. What have you learned? Now, for digestion, let's start with the carbohydrates. For the carbohydrates in the mouth, the saliva contains, what happens in the mouth? Because the saliva contains salivary amylase. What does this salivary amylase do? It will break down the starch, that is the carbohydrate, starch, into disaccharide or some monosaccharides. Does it make sense? Why in the stomach? Acid stops the action of salivary amylase. So this salivary amylase does not work in the stomach, only in the mouth. So there's no digestion in the stomach. So if they ask you, does carbohydrate digestion occur in the stomach? No. I need you to mark this. Let me mark it for you. You understand? Does it occur in the stomach? No. It doesn't occur in the stomach. But in the mouth, yes. Because there's no amylase in the stomach, salivary amylase in the stomach. All right? Now, let's talk carbohydrates in the intestine. What happens? Look at everything that happens. This is everything that happens. Remember, in the intestine, the pancreas secretes pancreatic amylase. Nothing happens in the stomach. but the pancreas stimulates pancreatic amylase. This pancreatic amylase helps to break down all these things. Examples of intestinal juice or includes maltase, lactase, and sucrase. Now let's see what they do. Maltase breaks down maltose to glucose and glucose. Lactase breaks down lactose to glucose and glucose. Sucrase breaks down sucrose to glucose and glucose. So all these enzymes are intestinal juices. You understand? They are intestinal juices. So they help to break down that carbohydrates in the intestine. So the breakdown of carbohydrates starts in the mouth, ends in the intestine, but nothing happens in the stomach. No this and no peace. Let's talk about the protein. For the protein, in the mouth, no digestion occurs. There's no protein digestion occur in the mouth. Remember I told you that chemical digestion of protein specifically starts in the stomach. But if they ask you, chemical and mechanical digestion, where does it start? Mouth. Why? Because chemical digestion of carbohydrates starts in the mouth. Salivary amylase. We discussed this some minutes earlier. You need to know the differences between these things so that you don't confuse them. But if they ask you in your question, where does chemical digestion of protein start? Please tell them in the stomach, not in the mouth. So no digestion of protein is found in the mouth. All right? In the stomach, what happens? You understand? We break down pepsinogen. This pepsinogen is inactive. It becomes pepsin. It becomes active. What does this pepsin do? The pepsin will now break down the protein to polypeptides. Make sense? Now let's see in the intestine. What happens in the intestine? What happens in the intestine? Digestion of protein in the small intestine. See, I just want to make this very brief because this video is getting too long and I really want to cut it very brief. What you need to focus your attention on, what is called proteases or peptidases. Proteases or peptidases. What do they mean? These are all enzymes that digest proteins in the intestine. These are enzymes that digest proteins, all right, in the intestine. Remember, in the stomach, we talked about the pepsin, correct? But here, we are talking about peptidases or proteases. What are they? Chymotrypsin, trypsin, peptidase. Chymotrypsin, trypsin, peptidase. What do? They convert all the proteins into tripeptides, dipeptides, and amino acids, which are the small parts of, the proteins. Do you understand? So polypeptide is converted to peptide. The peptide is converted to amino. So trypsin, chymotrypsin converts polypeptides to peptides. Then peptidase converts peptides to amino acid, which is this is what we are looking for. This is what is absorbed. Amino acid is the one that is absorbed by the intestinal villi. It's as simple as this. So just know these three. Chymotrypsin, trypsin converts polypeptides to peptides. Now, this peptidase converts the peptide to amino acid. That's all. That's all. That's for the intestine. And for here, we know that protein is converted by pepsin to polypeptide. So this polypeptide goes to the intestine and is converted, see, polypeptide converted by chymotrypsin and trypsin to this. And at the end of the day, peptidase converted this to this. So if they want to ask you the sequence, tell them that the sequence from the stomach is pepsin. After pepsin, what do we have? Trypsin, chymotrypsin. After that, what do we have? Peptidase. That is the sequence. You can't forget this now. I just helped you to break it down. For the digestion of fats, no digestion in the mouth, no digestion in the stomach. No this, no peace. They will ask you, one of the following does not have any digestion in the stomach. They are talking about fats because they are talking about fats. Also, remember carbohydrates, we don't digest it in the stomach. So the stomach digestion is usually what? If I go back to the slide, the stomach digestion is usually proteins. Fats, no digestion in the stomach. See? No digestion in the stomach because there's no salivary amylase. Okay. So going back to where we were, for fats, no digestion in the stomach, no digestion in the mouth, only digestion is in the small intestine. Let me break this thing down for you. Bile salts help emulsify fats. Emulsification. Do you understand? I don't want to go deep into it, but just know that emulsification droplets are broken down further by bile, lecithin, and agitation. What is agitation? Agitation is just usually things that are produced by segmentation. All right? So in the context, it's just like irritability, like discomfort, uncomfortable movement in the intestinal segmentation. That's what leads to agitation. So all these things helps to modify the fat droplets. Do you understand? Now, but just what you really need to know is that pancreatic lipase converts fat into fatty acids and glycerol. Pancreatic lipase. So when you are talking about fat, remember bile from the liver emulsifies fat. That's one. Remember things like bile, lecithin, and agitation produced by intestinal segmentation, all of them helps in emulsification of that droplet of fat. All right. So if you look at this diagram, you see fat, bile salts emulsifies fat. That emulsified fat is now converted to fatty acid and glycerol, which are the last component by pancreatic lipase. So this fatty acid and glycerol, what is now absorbed into the small intestine. All right. Another thing you need to know is about lipids. That lipids are transported to the surface of the intestinal absorptive cells by micelles and then processed into chylomicrons, which are secreted from the basal surface of the absorptive cells and taken into the lacteal in the villus. Why I read this part is that this part is very important. Read it 2 times or 5 times so that it will stick in your head. You know why? Because they may give you this thing and tell you true or false. They may give you this thing and give you options. But just know that lipids are being transported, you understand, to the surface of the intestinal absorptive cells by what? Micelles. So when they get there in that surface of the intestinal absorptive cells, what happens? They are now processed. So the lipids are changed to chylomicrons. You understand? The lipids are processed to chylomicrons and now taken into the lacteal of the villus, which we discussed before. So I need you to understand this. In terms of nutrient absorption, I won't say much, but just know that the process of absorption, in the intestine or the enterocytes is usually diffusion, osmosis, facilitated diffusion, and active transport. These 4 mechanisms is the way when they have been processed. Please remember this. This is the only thing I need you to remember from this particular part. Every other thing, we have discussed it. Every other thing. And another information you need to know is that vitamins, mineral salts, water, all these things, they are absorbed without digestion, direct. Remember I mentioned this thing initially. I'm happy that it came up here again, that the function of the intrinsic factor is that the intrinsic factor combines with the vitamin B12, which is ingested to bypass the stomach. Because if it's not combined with the stomach, what will happen? It will be broken down. That's why if people lack intrinsic factor, they come down with vitamin B12 deficiency. Another thing you need to know is that diarrhea occurs when large intestine absorbs too little water. You remember the large intestine has to absorb water. I mentioned it in the beginning of this thing. We call it compaction in the beginning of this discussion. All right. So the constipation is when there is too much absorption of water. That's constipation. So if you don't go to toilet or you don't poo for days or for weeks, maybe like 2 weeks, what happens? The intestine will absorb all the large intestine will absorb all the fluid from your feces. And what happens when it's coming out, it becomes so strong. So that's another thing you need to know. For the duodenum, we've already discussed the duodenum when we are discussing the small intestine. But the only thing I just need you to know is that it is C-shaped and it is the first and the shortest part of the small intestine. The duodenum, the ileum, the jejunum. Those are the parts of the small intestine. So it's just like we are doing the summary of the anatomy of the small intestine. Remember, the first part is the duodenum and is the shortest part. So if they tell you the duodenum is longer than this, tell them no, the duodenum is the shortest part. All right? And also it's the widest and the most fixed. It's the shortest, the widest, and the most fixed. We've discussed about this, the papillae of the duodenum when we were discussing about the gallbladder. We've discussed this, so you can go back and look at it. That's why I put this diagram. You understand? This is the duodenum. This is the hepatopancreatic ampulla, where the common bile duct and the pancreatic duct meet together, forming the hepatopancreatic duct and entering into the duodenum. I don't want to continue discussing it so that we don't waste time. The jejunum is just there. It just represents the proximal 2/5 of the small intestine. So the jejunum, takes over from the duodenum. All right. Now this is the ileum, makes the distal 3/5 of the small intestine. These are some things you need to know about it, which I want you to look at later. All right. Okay, so here is just a comparison of all of them in terms of location, walls, lumen, and mesentery. So go through it. Also in terms of villi, pairs, patches, and solitary lymphatic cells. Remember, remember the ileum has Peyer's patches. This is ileum, has Peyer's patches present, all right? So look at them. This is microscopic anatomy. I look at it at your leisure, all right? So now let's proceed to the last part, which is the large intestine. I know we've spent a lot of time, but please write down as much as you can. Remember as much as you can. You can watch this video over and over and over again for you to be well prepared for your exam in digestive system. Any college within the US or outside the US, you are welcome to use this video. If you are preparing for NCLEX, this is a perfect video for you, all right? Even USMLE, this is also a very good video for you. So it's good that you focus on the most important thing, because the problem we usually have as students is that we want to read everything. You can never know everything. So why not know the pointed informations? If you have a question, please put it in the comment section. Ask me a question in the comment section. Please leave please like this video and subscribe to my channel. So, and put the notification so that it can always come up when I post something else. All right. Now, the large intestine is approximately 1.5 meters in length, shorter than the small intestine. Know this. The large intestine is shorter than the small intestine. Large intestine is shorter than the small intestine. Even the fact that they call it large intestine. And there is no chemical digestion in the large intestine. There's no chemical digestion. These are the questions that they will ask you. Later, you can take your time to look at the anatomical sizes of the large intestine. We have the ascending, we have the transverse, we have descending, we have the sigmoid before we get to the rectum and the anal canal. These, their lengths, all right? So, another thing you need to know is the tenia coli, the hostra, and the epithelial appendages. We are going to look at it, but this is the anatomy of the large intestine. This is ascending, this is transverse, this is descending, this is sigmoid, all right? Usually your physics is stored here until this thing distends and you feel like pooing and you go and poo. If not, small intestine enters here. So by the time the food moves from here to here, the small intestine would have finished absorbing, rather the large intestine would have finished absorbing all the fluid in it. You understand? That is what we call compaction. So compaction specifically happens in the large intestine. So now when we are talking about the anatomy of the large intestine, we've discussed it initially. It has the muscularis externa, it has the omental appendages. Muscularis externa contains the tenia coli, hostra, internal anal sphincter, and external anal sphincter. Know the hostra very well, because the hostra pouches in the colon caused by the of the tenia coli. This is very important. Also know about the omental appendages, right? It doesn't have any known function, but it contains fatty pouches of peritoneum adhering to the colon. So you need to know these two things in the surface anatomy. Very important. Now, when we are talking about intestinal microbes and the gas that we pass out during physics, normally, first of all, we need to know the microbes, about 800 species of bacteria that populate the large intestine. Please know this. You must see it in your exam. Anybody asking you a question on the large intestine will definitely ask you about the microbes. Know that it has more than 800 or around 800 species in your intestine, all right? It has its own function. You know why? Because you cannot digest cellulose, for example. There are some vitamins you cannot synthesize, like B and K. You cannot also digest pectin. So this gut bacteria helps you to digest cellulose. And even there are some carbohydrates that we lack enzymes to digest. This is the importance of the bacteria. Don't think it's just for pathology. Oh, bacteria. No, this is very good bacteria, healthy bacteria. Some individual that lacks it, they lack all these things. The same thing with vitamin B and vitamin K, all right? Now, flatulence, that's passing gas, it's also a sign that you are alive. People think it's terrible. No, it's terrible, it smells. It's a sign that you're alive. That's why when somebody undergoes abdominal surgery, you understand, we wait for some time, for the person to pass flatulence to make sure that the gut is healthy, all right? So an average person passes about 500 mils of flatulence per day. And it smells. Why? Because it contains hydrogen sulfide, indole, and ketone. That's why it smells. Remember, this flatulence is usually as because of the air you ingested, or some of them because of those bacteria. So know this. These things are marked. The average person produces 500 ml. Very important. Why does it smell? Hydrogen sulfide, indole, and ketone. They will put it in the option. Flatulence smells because of the following, except they will put hydrogen sulfide, they will put indole, they put ketone, and they put something else. So remember, if you don't know it, if you don't know the physiology, you will not know the pathology. All right? Okay. Now, when you are talking about the absorption and the motility of the large intestine, just know that the large intestine takes time, about 36 to 48 hours to form physics, all right? And that physics that you are passing out contains about 75% water and 25% solid when it's produced. So if you don't go to toilet as at when due, you understand, the body will be absorbing back that 75% of water, and before you know it, you have a very strong physics. Now, that solid part, what does it contain? Please know this. 30% bacteria, 30% undigested fiber, 10% to 20% fats, small amount of mucus, proteins, salt, digestive secretion, soft epithelial cells. They may ask you, how many percent bacteria does the solid part of physics contain? Please, 30%. 30. 30% bacteria, 30% undigested fiber. How many percent water? 75% water. How many percent solid? 25% solid. Please know this. Very, very, very important. Another thing we need to know is the hostra. Now, I keep mentioning the hostra. What is even the hostra? Now, let me show you this picture. Look at this picture that we passed before. Now, remember we discussed about 3 things. We talked about the Taenia coli. This is Taenia coli, you see, this Taenia coli. That's why when I want to do you appendectomy, I just trace the Taenia coli. The place it ends, you see, is the appendix. I'll just catch the appendix. So that's the Taenia coli. Now, if you look at these things, that yellowish that falls out like leaves, all these things are what we call epiploic appendages. Epiploic appendages. So what is a hostra? Look at the hostra. You see how this place to this place is? That's a hostra. This is the where they label it. This is a hostra. So if you know the hostra, that's circulations of the wall, you know the Taenia coli, you know the epiploic appendages, then you are good to go. For real. You are good to go in terms of the absorption, motility. So why? Because the hostra contracts every 30 minutes. Remember, it contracts every 30 minutes. And when it contracts, what happens? It causes distension to that hostrum. You understand? Which will lead to, contraction. And this contraction leads to churning and mixing of residue, promoting water and salt absorption. So water and salt will leave. Every time it squeezes, water and salt will leave. All right? So I want you to look at this at your leisure, because this thing I really don't factor it in more. You understand? That's why I didn't. You can read it later. I just wanted to describe what the hostra does. So when you see this in your option, remember that hostra, the distension of the hostra leads to contraction, and this contraction leads to churning and the mixing activity of the water and the salt, thereby leading to increased absorption. All right? Now, so any minute you don't go to toilet, your salt and water is leaving your poo, thereby making it stronger. So this is the rectum, all right? This is the surface anatomy of the rectum. This is the divisions. Everything has been labeled. The rectosigmoid junction, very, very important, because we call it the watershed area, where it usually leads to watershed infarct. I'm not discussing this now, all right? So this is also the anal canal, right? So, the first part of the large intestine and of the alimentary canal, that is anal canal. This is anal canal. This is the rectum. This is anal canal here. So I need you to know that. It's the terminal portion, sorry, not the first portion, terminal portion of the large intestine. All right? So these are things you need to know about. This one continues about the anal canal, everything you need to know about it. Now let's talk about defecation. What leads to defecation? Stretching of the rectum stimulates 2 defecation reflexes. So what happens is the rectum, once it stretches, you want to, a reflex is activated. Now, what is that reflex that's activated? We have the intrinsic defecation reflex, which works entirely within mesenteric plexus to produce relatively weak response. All right? So usually the stretch receptors, they trigger the muscularis, leading to contraction, and the internal sphincter to relax. The internal sphincter had to relax. So this is the first part of the defecation reflex. Remember, defecation reflex. This accounts for the urge to defecate, often soon after meal. All right? The second one is the parasympathetic defecation reflex. This one involves the spinal cord. What does it do? Stretching of the rectum sends sensory signals to the spinal cord. When it stretches sensory signal to the spinal cord, this will increase peristalsis, thereby relaxing the internal sphincter. Relaxing the internal sphincter. So this is the internal sphincter, this is the external sphincter. If this thing contracts, you will be able to hold poo. That's why when you are in a place where you cannot poo, you will just naturally contract this internal sphincter and hold that poo for as long as possible. So usually when this internal sphincter, there is every possibility that you will rush to the toilet to poo. All right. So what these stretch reflexes do is just to increase the amount of peristalsis, thereby relaxing the internal anal sphincter. You're relaxing the internal anal sphincter. This same thing works here. So the system is just to relax internal anal sphincter. That's what defecation is about. Please look at these things properly. These are your questions. All right. Now, when you talk about the colon, we have the ascending, the transverse, the descending, the sigmoid. You can look at this later. Oh, this should not be here. Okay. So to round up the study, they may give you a lot of diagrams and they want you to label. So you need to know the most important part of those diagrams. So looking at this diagram, what can they point to here? They can point to this. This is the gallbladder. All right. Remember that this is the gallbladder. They can point to this. This is left lobe. This is right lobe. All right. The right is always bigger than the left. So you should be able to differentiate the two of them. All right. Let me see. So they can give you something like this. I'm pointing to esophagus, or point to the teeth, point to pharynx, or point to the oral cavity, or to the stomach, or to the liver. You need to know that this is accessory. They may tell you this, the liver, for example, produces what? Tell them that it produces bile. They may point to this. Just tell them that the function of this is to store bile. What is the function of pancreas? Pancreatic juices, all right? Large intestine, small intestine. So all these things has its own. I know that with this discussion, you'll be able to, dissect this diagram if they give you this diagram in exam, all right? Now, this is another thing that they can give you in exam. This is another thing they can give you in exam. This is a picture depicting the mucosa, the submucosa, and the muscularis. Please know this. I need you to know this sequence. They may point to this, tell them that this thing is in the muscularis, all right? So this is a plexus in the muscularis. What is it called? The mesenteric plexus. Muscular, mesenteric, muscular, mesenteric. Another thing for mesenteric is what? Albax. So this is submucosal plexus, submiasna. So another name for it is miasna plexus. You need to know this sequence. You need to know that from the inside to the outside is epithelium, lamina propria, muscularis, mucosa, then submucosa, muscularis externa, and serosa. If you're going from outside to inside, serosa, then in. You need to know this sequence. I've mentioned it again and again. All right? Now, this is the anatomy of the anatomy of the stomach, the gross anatomy of the stomach. All right? Remember that these are gastric food. They will ask you what is the importance of this gastric food. They will point to it. Or they may tell you that this part, when the peritoneum is attached to this part, where does it lead to? It leads to this is greater curvature, so greater omentum. Lesser curvature, lesser omentum. All right? So this is fundus of the stomach. This is the muscularis layer of the stomach. The muscularis layer of the stomach is 3. Longitudinal, external, circular layer, and the innermost oblique. These are very important. Remember that this is the pyloric orifice. You understand? This is pyloric sphincter that stops food from rushing into the duodenum, thereby giving time for the duodenum to function effectively. So these are the ways they can ask you questions on this diagram. This is another very, very, very diagram. Very, very, very important diagram that you need to know. This is the cephalic phase. What happens? You think about food, you see food, or you taste food. Everything starts working. HCl starts being produced in the stomach in expectation of the food. This is the second one, gastric phase. Food has entered the mouth to the stomach. What happens? There is still vagal response. You understand? This is the long reflex, sensory motor fibers, and all those things. While the short response is the mesenteric reflex. And what all these reflexes do? They increases production of gastrin and histamine. All right? Gastrin and histamine. Everything is in preparation of, in helping they're building a house for this food. Now, the last one is intestinal phase. In the intestinal phase, what happens? There is a reflex, So this reflex is as a result of food entering the intestine. Now, CCK is produced, secretin is produced, GIP is produced. All these things we call them intestinal gastrin also. All these things is for this food, to enter and to be digested properly. And also to inhibit the stomach. All these things will inhibit the stomach so that this place will not be open. The sphincter will be locked. You understand? Meaning that we want food to be digested in duodenum gradual. Now, among the second to the last diagram is the biliary system. What can they ask you here? They can point to this and ask you what is this. Remember, this is common bile duct, not common hepatic. This is common hepatic because it's coming from the liver, correct? It comes down here, joins with the cystic, and form common bile duct. So most times the stone will just block here or here. All right? But we are not discussing that now. This is the duodenum. They may ask you what is this. This is sphincter of Oddi. If they ask you what hole is this is hepatopancreatic ampulla. Extremely important. Why this is the pancreatic duct from the liver. All right? Now, the last part of this discussion is the gallbladder. They can give you this diagram. You understand? You need to know the diagram of the gallbladder very well because they can point to one or two things and ask you about it. They can point here and tell you what is this? What is the function of the gallbladder? The function is what? To store bile. What is the most important component of the gallbladder? Bile salts, which helps in the emulsification of fat in the intestine. Does it make sense? Good. So if you have any question, please leave it in the comment section. If you have a discussion, leave it in the comment section. We've been able to thoroughly dissect the anatomy and the physiology of the digestive system. Please, I need you to leave comments. Ask questions if there's something that I didn't explain properly. All right? Also, if you like the video, also make comments. Please, don't fail to like. Whether you comment or not, always click the like button. If you are not a subscriber, subscribe. Subscribe. Act on the notification. Do you understand? Show love. We took time to make this video work, all right? A lot of editing, a lot of talking, a lot of mistakes. But at the end of the day, we got it right. So I want you to be able to do it, to show appreciation by commenting, by liking, and by subscribing, on your notification. The next one that is coming is the neuro. Please keep in expectation. Thank you.